Hello and good evening to all of you. Am I audible? Okay, so today what we are going to do is yesterday we started with biotechnology, and today we'll complete or we'll try to cover the other dimensions of biotechnology. So today we'll be looking at application of biotechnology specifically in agriculture sector. So this application of biotechnology is also very important, and this year many of the GM crops were in news. So not only this GM mustard was in news, but also one of the GM rice was also in news, because some of the consignments in Europe that had reached from India, some of the consignments of rice. So the European authorities claimed that in the Indian rice was containing GM rice, and in some states, so in some states like Maharashtra, Gujarat, Telangana, Andhra Pradesh, it was observed that farmers were illegally growing another variety of BT cotton, that is known as HT cotton, and also cotton. So cotton, what you know that cotton is what we are saying, that it was introduced in 2002, and it has been 20 years now since BT cotton was introduced in India. So it has been 2 decades where cotton has been introduced. So with that respect, this application of biotechnology in agriculture sector is very important. So we need to know in biotechnology, how do we apply it in agriculture. So when we say GM crops, you need to understand that BT cotton, the purpose of BT cotton was what, that cotton plants, they are affected by pest attack. So in BT cotton, we'll see that what we have done is that we have made it pest resistant. But all GM crops will not be doing the same thing. So we'll be modifying the crops for different purposes. So in cotton, the purpose was to protect it from pest. But in, let us say, Dhara mustard hybrid 11, or your this GM mustard that we call it, the purpose was different. The purpose was to increase the productivity. So if you talk about BT brinjal, the purpose is different. Again, in BT brinjal, the purpose is very similar. It is also attacked by pests. So that is the purpose, that we need to protect it from pests. But if you look at, let us say, GM potato, so in GM potato, the purpose of going for genetic modification is different. The purpose there is to increase the nutritional value of potato, that so as to increase the protein content in potato. So in different crops, we are modifying the GM crop for different reasons. So in GM crops, you should always ask two questions: that why are we modifying the crop, and through what method. So that is a potential area of prelims question making, that why are we modifying it and how are we modifying it. So we'll look at some of the GM crops which have been in news this year. So this is a major application area of biotechnology in agriculture. So first of all, what we can do, we can begin with mustard itself, because mustard was in news. So let us just try to understand this mustard, GM mustard, then we'll come to other parts also. So GM mustard. So GM mustard was in news this year. So a GM mustard, it is also called as this. It is also called as your DMH11. So DMH11, DMH11. So why are we trying to modify mustard? So the reason for modification of mustard, we are going for modification of mustard, is, first of all, we need to understand that mustard is which type of crop. It is an oil seed crop. In mustard, what we are saying, that the yield has stagnated over the decades, and with respect to India, the oil, we are importing even vegetable oil. In fact, 55 to 60% of our oil demands are met by imports. So if we have to tackle that, we need to increase the production of the domestic oil seeds. So the major oil seed crops in India are your soybean, your groundnut, and then mustard. But in mustard, what we are seeing that the yield has stagnated over the years. Why it has stagnated in India? It is because of specific nutrients like boron, which is absent in Indian soil, and most of the regions where we are growing this mustard, it is dependent on a monsoon or it is rain-fed areas. Because of that, the yield has stagnated. And one of the biological reason which is cited is that GM is a self-pollinated plant. It is a self-pollinated plant. So what do we mean by a self-pollinated plant? It means that its flower, so its flowers has both the female part and the male part. So it has the anther. In the anther you will have the pollen grains, and it has the female part. So the female part, so there is this stigma. So when we say it is self pollinating, it means the pollen grains from the same flower, they are able to fertilize the ovum of the same flower. So because of that, what has happened is that the genetic diversity of this type of mustard had, excuse me, genetic diversity has drastically reduced. So because of self pollination, the impact is that the genetic diversity has decreased, and because of that genetic decrease in genetic diversity, the yield has reduced or dropped over the years. So what is the intention or what is the motive here, that how we can increase the yield? In order to increase the yield, we need to make it a cross pollinating plant. Cross pollinating, so we need to cross it with other varieties of your mustard. So what we have done in this case, so why is the GM mustard called as your DMH? So why it is being called as DMH 11? So this DMH 11, so first of all, some of the basics of this DMH 11, that it has been developed by Dr. Deepak Pental, who was Vice Chancellor of Delhi University, and his team. So they got funding from National Dairy Development Board and Department of Biotechnology also. So what they have done, they have utilized some genes from this bacteria. So here the bacteria is the name of the bacteria is Bacillus amyloliquefaciens. So this is the bacteria that is being used. So what we have done, that we have extracted some genes from extracted these genes from this bacteria, and we have transplanted it to this DMH 11. So let us just try to understand that what was done in this case, how we able to make this a cross pollinating plant. So we have taken why it is being called as a western variety. So we have taken western variety of wheat, and we have taken Indian variety of wheat. So Indian variety of wheat, and we wanted to cross it. So in order to cross it, we had to make its male part sterile. We had to make its male part sterile. How did we make the male part sterile? By adding this Barnase gene. So when we add this Barnase gene, the male part becomes sterile. So now the female part is ready to accept pollens from this western variety. So the pollen from this, it goes and it can pollinate this. So now what you get, the offspring that you get, that offspring is a hybrid. then what is the role of this gene, barster gene? So what happens is that this hybrid that you will get, the first generation, so let us say this is your first generation, this will also contain this gene, so its male will be sterile. So what we do is that we add in this variety itself, because we know that this variety will be having male sterility, so we are adding barster in this one. So this barster actually helps that the second generation now starts becoming fertile, or the male part becomes fertile, so now it can go for cross pollination after the hybrid has been formed. So in this way we have been able to get a hybrid. So this has been done in this case. What is the role of this bar gene? Bar gene makes your DMH11 what is known as herbicide tolerant. It makes the gene herbicide tolerant. So this herbicide tolerant, what do you mean by herbicide tolerant? So this is herbicide tolerant. Why do we need to make it herbicide tolerant? So that the farmers can apply herbicides. So what are herbs? So these herbs are unwanted plants, so we need to get rid of these unwanted plants. So to get rid of these unwanted plants, we add herbicides. So we want that we should not be applying this herbicide frequently, but at one instance only. So we are making it herbicide tolerant. And that is also one of the criticisms of this GM mustard, that we have added one gene which is making it herbicide tolerant, so that now the farmer will be incentivized to use more herbicides at one go itself. But the problem with that is that these herbicides are carcinogenic in nature. So environmentalists, they are concerned that if we go for GM mustard, then the farmers will be incentivized to grow or to put more herbicides, so which will eventually pass into soil, which will eventually pass into water bodies, and which can reach our food cycle also. So that is the concern of the environmentalist. And also one of the concern is that GEAC. What is GEAC? GEAC is the Genetic Engineering Appraisal Committee. So this body, GEAC, gave approval for seed production before commercial release. So GEAC has given approval for seed production, but in some cases, one case has been filed in Supreme Court, and Supreme Court has not given its decision whether we should allow GM mustard or not. So there was a criticism. So there is one body which is known as Coalition for GM Free India. Coalition for GM Free India. So they have criticized this GAAC for giving permission for seed production even when the matter is pending in the court. And they are also of the view that GEAC has not done exhaustive study on what could be the possible impact of this mustard on pollinators, especially the honeybees. So they have criticized GEAC. GEAC is the regulator of biotechnology in India. GEAC is the regulator of biotechnology in India. And sometimes you will see that it is attracting some criticism. So in the last part we will also try to understand what is the role of GEAC. Is it a statutory body or not? Okay, so a simple question: is GEAC a statutory body, or is it not a statutory body? Is GEAC a statutory body? So these are your prelims level questions only. So they have asked, UPSC has already asked questions based on this understanding, that GEAC is a statutory body or not. If it is a statutory body, it works under which law, or is it working under any ministry? Yes, anybody would like to try? Is GAC a statutory body? Okay, let me just bring that question that was asked. The Genetic Engineering Appraisal Committee is constituted under which act? Yes, give it a try. Okay, so the Genetic Engineering Appraisal Committee is constituted under this Environmental Protection Act. Environmental Protection Act. So this was a question which has already been asked, so they can ask a higher level of question. So yes, it is a statutory body, and it operates under this Environmental Protection Act, EPA Act of 1986, and it is also governed by what is famously known as the 1989 rules. 1989 rules. So we'll come back to this GAC, and we'll see that there are some criticisms of GAC that is important from a mains point of view, but we'll just look at some of the criticisms of your GAC also. So coming back to your GM mustard, so what are the advantages of GM mustard? So we have this Dr. Deepak Mental and his team. So there is a wing of your Delhi University. So that research wing has actually developed this DMH11, and GAC wants to give approval for commercial release also, but there are criticisms by different agencies, so it has not been allowed. So GM mustard, if it gets a permission, it could be the first GM food crop of India. It could be the first GM crop of India if the Indian government gives its permission. But till now, it has not given its permission. So, what are the advantages of going for GM mustard? So, what we have seen that the yield has increased in so yield has increased to around 28 to 30%. So the yield has increased, and we have also seen that the nutritional value is also it has also improved. So the nutritional value has also improved of this GM mustard. So that is an unintended benefit that we have seen, and because it is herbicide tolerant from farmers point of view, so growing these crops would be easier. Why? Because they can spray the herbicide at one go, so it will reduce the labor cost of the farmers. And one of the biggest advantage is that since it has this seed has been developed by government agency, so we'll see that in case of bt cotton, so there is this company which has made this seed, Monsanto. So have you come across this company? So Monsanto has made this bt cotton seed, and they have produced what is known as suicide seeds. Monsanto has made BT cotton seeds as suicide seeds. So, do you know what are suicide seeds? What are suicide seeds? What are suicide seeds? So, what Monsanto has done is that they have added some genes deliberately in the BT cotton seeds so that their next generation is not fertile. So, what the farmer has to do, farmer after every crop cycle, they have to buy new seeds. That is the biggest problem with respect to BT cotton. So, farmers are forced to buy new seeds, that increases their labor cost, and it also often leads to exploitation of the farmer. But in case of GM mustard, it is not developed by a private company, but it is developed by government body. Since it is developed by government body, it will not be a terminator seed or not a suicide seed. So a farmer need not buy these seeds again and again. So that is also a biggest advantage from farmer point of view. And with respect to India, what is the advantage for India? So we see that 55% to 60% of our vegetable oil needs, we have to meet it by imports only. So 55 to 60% is met by imports. And if you look at the stats, it costs India around rupees 1,20,000 crores per year. That is a huge amount that India pays to import vegetable oil. So if we can increase the productivity or yield of GM mustard, this import bill of India can also drastically reduce. So that is the objective, that is the purpose of GM mustard. But what we have seen, that the yield is improving, the nutritional value is improving, the labor cost of the farmer is decreasing, and lesser exploitation of the farmers, because these seeds are not suicide seeds, and it can for the country, it can reduce the import bill also. So that is why there are some strong groups even among the government and among the farmer groups that want that this GM mustard should be made legal in India. So we'll wait and watch, and we'll see that when this happens. But if this happens, then this GM mustard could be the first food crop which could get legal permission. So in India, you should remember that there is only one legal GM crop. So there is only one crop which is a GM which is legal, that is only BT cotton, and no other type of GM crop, not even HT cotton is legal in India. So we'll try to understand what is this HT cotton also. BT cotton also will try to develop an understanding, but you should remember that only legal crop in India is BT cotton, and no other crop is legal in India. So you should also remember that when we say GM mustard, the bacteria name is not very important, but you should understand that this is not same as the bacteria as it is used in cotton. So in cotton, what we are using Bacillus thuringiensis. So that is a common type of bacteria found in soil. So this bacteria is a different bacteria, and we have extracted the genes from this bacteria, and we have made this this hybrid variety. So let us now look at cotton, BT cotton. So any doubts till now? Ram, if you have any doubt, you can ask. Okay, so see, last year, let us see the question that was asked in 2021. So what was the question that was asked in 2021? So let us give it a try. So Bolgard 1 and Bolgard 2 technologies are mentioned in the context of. Yes, give it a try. Bolgard 1 and Bolgard 2 technologies are mentioned in the context of clonal propagation of crop plants, developing genetically modified crop plants, production of plant growth substances, production of biofertilizers. Yes, anybody would like to try? Give it a try. Okay, so the Bolgard 1 and Bolgard 2 technologies are mentioned in the context of your clonal propagation of crop plants, genetically developing genetically modified crop plants, production of plant growth substances, production of biofertilizers. So this is about genetically modified crop plants. This is specifically about BT cotton. So in BT cotton, we have developed BT seeds, or what are those seeds are called as Bolgard seeds. Bolgard, these seeds are protecting against bollworms. So cotton plants are attacked by what are known as bollworms. Are attacked by bollworms. So we'll try to develop an understanding of this BT cotton itself. Why BT cotton is the only GM crop which is legal in India, and it was made legal in India in 2002. So it has been 2 decades now, and we there could be a potential prelims question also. There could be a potential mains question also with respect to our understanding of cotton. So we'll try to develop an understanding of this cotton. So in order to understand this cotton, why did we go for BT? So first of all, that we had said in the start also, that what are we going to do. So we need to understand that why the modification was done, and then we'll see how the modification was done. So we need to understand why the modification was done. So in the left hand side, that you can see that healthy this is a healthy cotton plant. So this is a healthy cotton plant, and in the second one that you can see here, so you can see that there is a big hole made by the pests, and it is destroyed. In the third one, you can see again, this is also a completely destroyed crop cotton plant. So pest attacks severely cotton plants. Because the pests attack severely the cotton plants, we need to protect cotton plants from these pest attack. So what was done traditionally is that we used to spray some pesticides. So we used to spray some pesticides to protect our cotton plants. But this pesticide, the problem with the external spray pesticide, or which type of pesticide was used, even the pesticide that we were using, we'll see. So what in this slide you can see that the cotton plant is attacked by more than 162 species of insect pests. The insect pest. So there are many in pests. So there are many complexes that we call these pests, a group of pests, as your complexes. So you have the sucking pest complex, then you have what is known as bollworm complex. So in bollworm complex also, you will see that there are 3 types of bollworm. There is this American bollworm, there is this pink bollworm, there is this spotted bollworm. This American bollworm is the major one. So it attacks the 80% of the crop plants in India. So what were we doing before BT cotton to protect these cotton plants? So in that scenario, what we were doing was that we were spraying with some pesticides. So we were spraying pesticides. So this pesticide was derived from a bacteria known as BT bacteria, or Bacillus thuringiensis. This bacteria, if you see, this bacteria produces what is known as some crystal protein. This bacteria produces crystal protein. This crystal protein actually a crystal protein in inactivated state. So this is inactivated crystal protein. When your insect, if you're spraying, let us say, these crystal proteins, and when the bacteria, oh sorry, not the bacteria, when the pests are eating it, so let us say the pest eats these crystal proteins. So when the pest eats this crystal protein, what eventually happens is that these crystal proteins inside the gut, they get activated. So these crystal proteins, they get activated and they become like this, or crystalline structure inside the gut. Inside the gut means inside the intestine. So inside the intestine there is high pH, or there is a basic environment inside inside the gut. So these crystal proteins, they get activated and they actually rupture the, so these crystal proteins, they rupture the gut of the pest. They rupture the gut of the pest, and because of that these pests die. So these pests die. But when we go for external sprays, what is the problem that we see? So when we go for external spray, the problem that we see is that crystal proteins these crystal proteins, so when we are using these sprays, what are the limitations that we are seeing? That the toxin is rapidly degraded in ultraviolet light, or in high leaf pitch, or desiccation. So in very dry environment it gets lost, and caterpillar must eat enough treated plant tissue. So these insects should eat a lot of the plant tissues where there is spray. So only if they eat in sufficient quantity will they die. So that was also a concern, and the site where feed are difficult to cover with external sprays. So what we are saying here, that because of the external sprays there are some parts of the plant which we are not able to reach, and these pests will grow there. So because of the external spray, this is the problem that we are facing. Another problem that we face when we go for external BT sprays is that the older larvae, the older insects, they are not affected by this, or they somehow have become immune to this. So in order to tackle these issues, it was thought that we will introduce the gene which is helping the bacteria to make these crystal proteins. We will introduce these genes in the plant tissue itself, so the plant itself will make these crystal proteins. So what we are doing here, that instead of going for external spray, we are trying to develop a mechanism in which we will do what, that these crystal proteins, this crystal protein, this we will not spray externally, rather this crystal protein, we found that there are some genes which are responsible for making this crystal protein. So what we are doing is that we will extract this gene. We extracted the gene and we inserted in cotton plant, and we inserted it in cotton plant. So because this gene is now inside the cotton plant, your cotton plant is also able to make crystal protein. So your cotton plant is making crystal protein, and that crystal protein, when your insects or your larva, they eat these cotton plants, so they will face the same scenario, that these crystal proteins get activated inside the gut of the insects, and it ruptures the guts of the insects. So that is how they get killed. That is how the pests get killed in case of BT cotton. That is why this is important. So you should understand that this BT cotton is in news with respect to one of the incidents, that farmers in some states, they were growing what is known as HTBT cotton. HTBT cotton. So before that, what is this HTBT cotton? Let us understand that when we introduce it inside the plant tissue, when we introduce the gene inside the plant tissue, what are the benefits? So the benefits are that it is not getting rapidly degraded in the environment, and also the coverage, there is no coverage issue. So this gene is present in all the parts of the plant, this protein is present in all parts of the plant. So the larva, whenever it eats anything from the plant, will eat the crystal protein, and eventually it will die, and there will be less incidences of larva becoming mature larva or adult larva. Why? Because at the very young age itself, they will start feeding on the plant and they will keep dying. So they will not eventually become bigger larva. So we were seeing that the bigger larva were becoming immune to this, so that situation is also tackled. So that is the advantage of introducing the gene into the cotton plant itself from the bacteria. So that is how it provides immunity. So this bt cotton this bt cotton was first introduced in USA. So in USA in 1996-97 time, it was first made legal. So in USA it was first made legal. So this company in there were three companies actually. So Monsanto, Delta Pine, they developed this bt Delta Pine. They developed this bt cotton, and during the mid of 1980s, so 1980s, mid 1980s, they developed this, and then it became legal in USA. Then in India in 2002, GAC gave approval. So in India, GAC gave approval gave approval. But you must take a note that cotton is a non-food crop. Cotton is non-food crop, and till now we haven't given any permission to any of the food crops. So since it was non-food crop, maybe we have gone for giving permission to this plant, but we haven't gone for giving permission to food crops. So what are the advantages of what are the advantages of bt cotton? So in this slide you can see that there are many advantages of bt cotton. So when we went for bt cotton, it means that when we introduced the gene from the bacteria into the cotton, so there were many advantages. So now we needed less pesticides. We needed less pesticides, and because of less usage of less pesticides, so you see there are many benefits. So what are the different benefits? So first benefit that you are seeing is that the reduced pesticide usage, improved crop management effectiveness, so reduction in production costs. So if the labor cost is reduced, the pesticide cost is reduced, and it is improving the yield and profitability. So that these are some of the direct benefits. And obviously let us see the indirect benefits of bt cotton. So it improves the population of beneficial insects and wildlife in cotton field. Why? Because if we are using less pesticides, even the beneficial insects will survive. In earlier cases, if we are using too much of pesticides, even the beneficial insects were getting lost. That was a major concern. That is, the if we start using too much pesticides, then we'll lose out these beneficial insects. And because we are not using pesticides, or there is very less usage of pesticide, so there is no issue of pesticide runoff. So pesticides will not percolate in soil, it will not run off into water bodies, so your water bodies are also safer, and then it will not enter the food cycle. That is how it will protect or save us from this type of problems. So air pollution and waste from the insecticides. So air pollution, when you are spraying these insecticides, so it becomes a very small droplet type of structure, what is known as aerosols, and we often use some machinery to spray this, so that also leads to burning of fossil fuels also. So we'll be using less fossil fuel, there will be less air pollution. Will it improve the farmers' health also? Yes, the farmers will also be less exposed to usage of pesticides. So because we are using less pesticides, so farmers' health is also ensured, and environment health is also ensured. The other benefits, that is that if the yield is improving, so there will be surplus. If there is surplus, then the Indian farmers can go for export. So because of export, the farmers income will increase. So if the farmers income if it increases, then overall it benefits what the growth of the country, or the GDP of the country will also rise. So these are some of the benefits of this bt cotton. So it has been 20 years now since we have started growing bt cotton. So when we started bt cotton in 2002, so of all the total cotton crops that were grown in the cotton fields, only 1% was bt cotton. Only 1% of all the total cotton fields, 1% was bt cotton. But by 2022 we see that 96% of all the cotton acreage or cotton field, we see that 96% is bt cotton now. So in 20 years you can see the increase in the acreage or area of the cotton plant. So your bt cotton has become the dominant cotton. So we are this brings us to the negative aspect also of growing bt cotton. So we'll see some of the challenges or concerns with respect to bt cotton also. So what are the concerns that we are seeing with respect to bt cotton is that there is rise of secondary pests. So what is this secondary pest? So in the first slide that we, in the first few slides we had seen that the cotton plant is attacked by many types of complexes. So there is sucking complex, there is this bollworm complex. So the bollworm complex is the main pesticide which is attacking the cotton plant. So what we are doing here is that since we have got rid of the bollworm, now the sucking complex, they are taking the primary role. So they are becoming very prominent now in attacking cotton plant. So we have got rid of one problem, but the other problems are arising, and what we are seeing that there is a resistance built up towards bt. And what has happened is that natural parasitic enemies of the cotton bollworm has also decreased. So that is the problem that we are facing. And even these bollworms are also reacting, and they are becoming resistant to these bt. So that is why we need to keep on evolving, and we need to keep on modifying these bt cotton. So you would see that we are developing what is known as the second generation of bt. So second generation of bt, so here you are seeing, so we have added not only one gene. In the first generation we were adding only one gene, but in second generation you can see that we are adding one more gene. So these are also the different bt cotton, only with different variety of bt cotton. So you're seeing here that we have to go for what is known as a second generation, then we need to go for third generation. So this is the Bolgard 2, that is the second generation. So in third generation, when the pests will become more resistant, we'll need to add more genes. So we'll need to add genes not only from one bacteria, but maybe from other sources also. So that is a problem that we are seeing, that bt is becoming, or these bacteria these pests are becoming resistant. And one of the biggest problem or concern with respect to farmers was this monopoly of seeds by these seed companies, by seed mncs. So Monsanto has monopoly over your bt cotton. So Monsanto has a monopoly of bt cotton, and because of that, so what they have done is that they have made the bt cotton seeds as terminator seeds. So what are these terminator seeds? So they have added some genes in the bt, in the in the crop plant, so that the second generation is infertile. Second generation is infertile. Yes, hello Manvendra. So what they are doing is that they have added some genes to make these seeds of bt cotton as terminator seeds, or what we call as suicide seeds. So because of that, the farmer cannot use the seeds of the parent crop to grow for next year. So farmer always has to go to buy these new seeds for every crop cycle, and because of that we are seeing the exploitation of the farmers, or farmer has to pay premium for these type of cotton plants, and that is a concern. And also what has happened is that we have lost, or we have decreased the genetic diversity of the cotton plant. We have decreased the genetic diversity of the cotton plant. So you would have seen that the cotton of all the cotton area now 96% is bt cotton only. So if we are growing so much of bt cotton only, it means that we have lost the indigenous varieties. Lost the indigenous variety. So because of that, there are concerns that what in future if we lose out on the most of the genetic diversity. So in future we will become, or these cotton plants will become more vulnerable to pest attack. सर आपको बहुत दिन के बाद देखे लगता है. Okay, so I have yes, online my presence is little less, that I realize, but I am quite regular in offline. So I teach quite extensively in offline, so I have quite exhaustive classes in offline. But online, yes, I'll try to increase my presence here also, so that it benefits some of the aspirants, especially those who are giving prelims this year. Otherwise we'll keep on coming up with more such videos. So this year in this bt cotton is important not only from mains point of view, but it is also important from prelims point of view. So you need to have a good understanding of bt cotton. So why we have gone for modification of cotton, it is because it gets attacked by pests. So we have made it pest resistant. And how we have made it pest resistant? By adding some genes from a bacteria which is known as bacillus thuringiensis. And we have seen that because we are adding those genes, so there are many advantages that it has got. So let us move now to what is known as HTBT cotton. So now this question would be easy to you. So Bolcard 1 and Bolcard 2 technologies are mentioned in the context of, so this is mentioned in the context of BT cotton. This is mentioned in the context of BT cotton. Okay, so let us try to understand this HTBT cotton. This HTBT cotton was in news, so you should be knowing about this HTBT cotton. Why this was in news? It was in news because some of the farmers they were found that they were growing illegally HTBT cotton. So HTBT cotton is, again, yes, it is a BT cotton, but it is not legal in India. So only legal crop in India is your BT cotton and not even HTBT cotton. So what is this HTBT cotton? So HT means here herbicide tolerant. HT means here herbicide tolerant. Herbicide tolerant. Herbicide tolerant. So what is this herbicide tolerant variety of cotton? So cotton farmers, they have to spray herbicides. So what are herbicides? These are chemicals. Herbicides are chemicals which are added to kill weeds. Weeds are your undesired plants which grow along with, let us say in this case, the cotton plant, and we want to get rid of those undesired plants. So we use herbicide. And why are we trying to make herbicide cotton as herbicide tolerant? Yes, so that when we add these herbicides, so it kills the unwanted plants and not the cotton plant. And also one of the reason here from the perspective of farmers is that farmers have to spray these herbicides often, or at least twice during the crop cycle of cotton. What they want is that if the cotton plant is resistant to herbicide, then they can add add herbicide at one go itself in large quantity, so that they do not have to spray it at regular intervals. So that is why they want it. But what is the concern? Is the concern is that these herbicides are carcinogenic. So these herbicides are your glyphosate, and they are carcinogenic in nature. So because of that, there is criticism that we shouldn't go for this HTBT cotton. So uh, so the modification here is that we have made it herbicide tolerant, but it is illegal in India. So the only legal crop that you should remember is BT cotton. Even HT BT cotton is not legal in India. In this, there is a term which is called as super weeds. What do you understand by this term, super weeds? Yes, anybody would like to try what are super weeds? Or you would hear this term super bugs also. What are super bugs? What are super bugs? Yes, anybody would like to try? BT cotton is legal. BT cotton is legal, but any other crop is not legal. Only BT is legal. Even HTBT is not legal. HTBT cotton is also not legal. Only one crop is legal in India, that is BT cotton. Is it clear, Aditi? Okay, so insects. So when you say super bugs are what? They are insects only which have become resistant toward all these manipulations that we have done. So it has become resistant to crystal proteins. Super weeds are your plants. These are plants or weeds which have become resistant to these herbicides also. So those are super weeds. So herbicide resistant weeds are your super weeds. So if we start using too much of herbicide, then slowly and slowly you'll see that even the weeds will become resistant. Just as in the case we were saying that this in case of cotton, we are saying that resistance is building, so it is leading to formation of super bugs. It is leading to formation of super bugs. If in this case what we are saying, that if we start using too much herbicides, then what will happen? Even the weeds will become resistant. Even the weeds will become resistant to herbicides. And that is what we call as super weeds. Okay, we'll come to this flavor saver tomato a little later, but let us see some of the other important GM crops. Yes, so since we have done the GM mustard, let us try this question. Sir, reason for illegality is carcinogenic nature or these anything else to it? Sorry, sir, typing took time. See, uh, there are not just only one of the reasons. So there are many such concerns that if we open this Pandora box of allowing more GM crops, then there could be demand that we should allow BT brinjal also, GM mustard also, or GM potato also, GM rice also. So from a governance point of view, they are concerned that people would demand more. So that is why they are stopping it. But one of the major reason is yes, because it is carcinogenic. So they government or different uh, groups, they do not want that we should be using HTBT cotton. Yes, sir. Please try this question. So we have already discussed this GM mustard. Just give it a try. D. We had said, see why it is not D. It is not D because this statement is not correct. Because this has been not developed by Punjab Agricultural University, but this has been developed by Dr. Deepak Pental and his team at Delhi University. So Dr. Deepak Pental was Vice Chancellor of DU, and he in DU there is a research wing which has developed this GM mustard. It is not this organization. So third is not correct. Why do you think this one is also not correct? Is the first statement correct or not? Is this is this statement correct? This statement is not correct. Why? Because the objective of modification of GM mustard is to not make it pest resistant, but to make it what to increase the yield. So to increase the yield, we have added genes from different bacteria. We have not. The purpose of GM mustard is not to make pest resistant. Pest resistance is for cotton. Pest resistance is for cotton, not for mustard. So only your second statement is correct. So that is what I am saying, that you should develop an understanding that when we are going for genetic modification, it doesn't mean that since we have gone for making cotton pest resistant, it means that all the GM crops that you will see, they will be we are modifying it to make it pest resistant only. So the objective is different in a different plant. So if you're talking about mustard, so it is different. If you're talking about brinjal, the purpose is different. If you're talking about modification of rice, purpose is different. Okay, so it is not that we are doing the same thing in all the GM crops. So we are doing different things in different crops, that we'll see. Now this BT brinjal also keeps coming in news. So this BT brinjal also you should know why, because in 2007 itself, in 2007 itself, GEAC had given approval for its commercial release. GEAC had given approval for commercial release, but however, government at that point of time, it faced criticism and opposition, so it did not allow BT brinjal. Had this been allowed, so it would have become the first food crop which would be a genetically modified crop, and this would have been allowed in India, or which would have been legal in India. But even BT brinjal is not legal in India till now also. So what are we doing here? So again, we are using the same bacteria that we have used in cotton. So we are using the Bacillus thuringiensis, and again the problem with brinjal also is similar only. So it gets attacked with pests. Because it get attacks with pest, so those pests, they eat away the fruit, or they eat away the shoot. So those pests are, so the scientific names are not very important, but it is Helicoverpa armigera. So there are shoot borers, so what we have done is that again we have extracted this particular gene from BT, and we have inserted it into the brinjal plant. So we have extracted this from the bacteria, and we have inserted it into the brinjal plant. So in that way we have been able to make it resistant to the pest attack. And what you should note here, that this particular BT brinjal is not made by a private company, but these are your government organizations. So it has been made by a collaboration of your Mahiko, that is Maharashtra Hybrid Seed Company, Dharwad University of Agriculture Sciences, and Tamil Nadu Agricultural Universities. So there is no monopoly of a private company. So these seeds have been developed by government agency. So again the benefit here is that farmers will not be exploited, just as in case of BT cotton. So GAC had given permission, but what had happened, that government blocked it because it faced opposition. In Bangladesh, however, Bangladesh started growing this BT brinjal. Because it started growing this BT brinjal, so it has been a success story in Bangladesh, and because of that, so Indian farmers are also demanding that the government should allow growing up of your BT brinjal. But India is not allowing the growing up of BT brinjal. That is why you should know this, that it could have become the first food crop in India had it been made legal way back in 2007. So so you can see in this picture that BT brinjal has been allowed in Bangladesh since 2013. So these are some of the varieties of the BT brinjal. Now this GM rice is also important from exam point of view. So why this GM rice is important from exam point of view? So it was in news. So it was in news. Why? Because some of the rice which India had exported to Europe, European authorities said that, or they blamed that in those rice there was GM rice. But in India, GM rice is not legal. So however, government denied it. And which type of GM rice was found? So golden rice was found. Golden rice was found. So what is this golden rice? We need to understand this golden rice. So what are we doing in this golden rice? So this is how the golden rice looks like. So this on the left hand side, what you can see is this golden rice. This is the normal rice. So why it is golden in color? It is because it has provitamin A. It has some provitamin A in it, and provitamin A means that it has some immature vitamin A. If we consume this provitamin A inside our body, it will convert into vitamin A. So that is why. So this vitamin A is present in your carrots or other leafy vegetables which are orange or yellow colored. So that is why this rice is also this orangish or yellowish in color. So what we have done here is that we have increased the nutritional value of rice. So in golden rice, the objective is to increase the nutritional value of rice. The nutritional value of rice. So how we are doing that, we'll see. But what we are doing is that we want to increase vitamin A in rice. And how do we do that? We'll see. So what happens in normal scenarios, that in rice, so if you see this type of rice, so in this rice plant, in every part of the rice plant, we see that vitamin A accumulates. Vitamin A accumulates, but only in the seed vitamin A does not accumulate. So there is some sort of gene silencing which happens in the rice seeds. So what we have done is that we have extracted some genes and we have added those genes to the rice plant, and now these genes are helping to accumulate even vitamin A inside the seeds also. That is why the plant is able to add vitamin A into this. So this is also known as what? Biofortification. Biofortification. So this process is also known as biofortification. Why it is known as biofortification? First of all, what is fortification? Anybody, what is fortification? What is fortification? What is fortification? What do we understand by fortification, this term? What is fortification? Give it a try. Adding minerals to rice, yes. So adding minerals or vitamins to rice, strengthening. Fortification means, okay, so we are talking about the literal meaning of the word fortification. So yes, there are political fortification also, and here when we say fortification, here we mean specifically that we are adding nutrients to strengthen, to make strong. Okay, yes. So even in salt, we add what? Iodine to make it fortified. So it has iodine to make it fortified. Similarly, what we are doing here, we are going for not external addition of nutrients, we are going for internal addition of nutrients. That is why we are calling it as biofortification. So it is slightly different from external addition. So the plant itself is adding the nutrients. So in this case, the plant itself is adding vitamin A. That is why it is called as biofortification. So in golden rice, it is a new type of rice that contains beta carotene. Beta carotene is your provitamin A, which gets converted into vitamin A on eating. So beta carotene is found in leafy vegetables, orange and yellow colored fruits. Principal use, we have said that in rice plants, they synthesize rice plants synthesize beta carotene in all parts of the plant except the grains. So two genes are added and the pathway is switched back on, and consequently beta carotene accumulates in the grain itself. So this GR2, so this golden rice have the same yield and agronomic performances as their conventional parts. So what we have seen, that we add genes to switch back this pathway on, so that beta carotene starts accumulating even in the rice grain itself. So what are the advantages of golden rice? Golden rice, vitamin A. What is the significance of vitamin A in our body? Vitamin A is also known as, the scientific name of vitamin A is retinol. If there is deficiency of vitamin A in our body, so it leads to which type of disease? Deficiency disease. Yes, so lack of vitamin A or deficiency of vitamin A leads to which type of disease? Yes, anybody? Hair loss. Vitamin A. Hair loss is a complex phenomena. It is caused by many things. Hair loss is essentially, or you can mainly attribute to deficiency of vitamin E. So hair loss is caused by your vitamin E mainly. Yes, Aditi, night blindness. So xerophthalmia. Xerophthalmia is dryness of eyes. So it leads to dryness of the eyes and the cornea, and it leads to what is known as night blindness. Blindness is called as nyctalopia. And because of deficiency of vitamin A, our immune system also gets weakened. Immune system gets weakened. So vitamin A is a major concern, or deficiency of vitamin A is a major concern for developing countries and poor countries, or African countries, or the Asian countries. So in Africa, the children, they suffer from this deficiency, and they suffer these problems of xerophthalmia, nyctalopia, that is night blindness, weak immune system. So in Africa and Asia, this is a big or a major concern. So that is why we are trying to develop golden rice. So in golden rice, we are going for biofortification. So biofortification, through biofortification, we'll be able to add this vitamin A, provitamin A. Why are we using this term provitamin A? It is not mature vitamin A, but when we eat, when it enters our digestive system, then from provitamin A it becomes your vitamin A. So provitamin A inside our digestive system will become vitamin A. And this golden rice, the benefit of the golden rice that you can see is what it is saying, that just consumption of 100 to 150 gram of rice can help in meeting the 60% of the daily requirement of vitamin A. And see, getting vitamin A sometimes becomes difficult, so that it can address this situation. What it is also saying that it will reduce the gastrointestinal distress. So vitamin A, it helps in good digestion also. That is what it is saying, that it will help in good digestion or reducing the gastrointestinal distress. And since the golden rice is low fat and low sodium, so it is good for lowering the BP and cholesterol also. So it will be good for heart patients because it is containing low fat and low sodium. And what we have said, that vitamin A deficiency is a major problem in developing world, causing blindness and impairing the immune system, particularly in children. So this is a cause of concern in developing world, and specifically among children. So golden rice could be a boon in such scenarios. Okay, so let us see what are the concerns with respect to this GM rice or golden rice. So one of the philosophical concern is that we are using genetically modified rice to improve human health. So for improving human health, we need to improve our diet and improve our food. But there is a perception that if we are going for genetic modification, why do we need to go for genetic modification? It is because of the previous mistakes that we have made with respect to green revolution, that now we have food sufficiency, but the quality of the food or the nutrition level of the food has degraded, and because of that we are now again trying to modify the crops. So that could be problematic in the future. So that is leading to these type of perception issues. And what it is also saying, that in the initial trials of the golden rice, it did not contain enough vitamin A. It did not contain enough vitamin A. And the nutrients to which nutrients persist once the rice is has been cooked is not very clear. So with temperature, we do not have a clear picture that whether these vitamin A will still remain in the rice, or because vitamin A, because of heat, it might break down or it might not pass on to the body. So these are some of the concerns with respect to your rice. This GM potato was also in news. So GM potato is known as potato. Potato. Why? Because it is containing protein. So the potato that we eat has very less proteins, and we want to increase the nutritional value of the potato. So we have added some genes from a plant known as amaranth, from a plant called as amaranth, and from that we have taken what is known as AM1 gene, AM1 gene. So we have inserted this AM1 gene in potato, and now what we are seeing, that the protein content in this potato is increasing to more than 60% compared to normal potato. So there is higher protein in your potato. So it has been developed by there is National Institute of Plant Genomic Research, NIPGR in Delhi, and there is Central Potato Research Institute SIPRI, that is in Shimla. So they have developed this GM potato. GM potato. So what we are seeing, that it can become a good source of proteins along. Yes, potato, that we know, it is a good source of carbs, but we, in our wholesome diet, we also need protein. So what we can do is that we can add protein even in this potato. So again, this is also an example of biofortification. This is also an example of biofortification. So let us look at these rice genome projects. Not very important from exam point of view. Why? Because these are some of the old projects. But today on my channel I have shared some article with respect to sequencing. Sequencing, let us just try this question. So what do you understand by sequencing? Let us just try to develop this understanding. Sequencing, this concept is important for prelims this year, so you should be understanding what is sequencing. What do you mean by sequencing? Just give it a try. Yes, there is some lag with the internet, I guess. So, sequencing is mixing up of different genes. That is not quite correct. Sequencing is not mixing up of genes. So mixing up of genes is called as what, recombinants. So recombinant DNA or genetic engineering. What is sequencing? Or try to answer this question, then we'll see. Yes, anybody else wants to take a try? Okay, so let us just read this question. With reference to agriculture in India, how can the technique gene orientation in different pattern? Yes, so sequencing. So yesterday we were discussing about these base pairs. What are these base pairs? So our DNA is made by your base pairs only. So your DNA has many base pairs. So sequencing means trying to find out the exact pattern of these base pairs in humans, in plants, in animals. So here when you say about genome sequencing, it means that you're trying to find the sequence of the base pairs of the DNA, and you're trying to locate some of the genes. What are genes? Genes are a functional part of the DNA, and these genes would contain hundreds or more than hundreds of the base pairs. So you're trying to find the location of the genes also, and you're trying to find the exact pattern of the base pairs also. That is what is known as sequencing. That is what is known as genome sequencing. So genome sequencing can be used to identify genetic markers for disease resistance and drought tolerance in various crop plants. Yes, so genome sequencing can be used to identify genetic markers for disease resistance. So there could be that in plant, because of the presence of some particular kind of gene, so it is becoming resistant for disease and drought tolerance in various crops. So we can with that by sequencing only we can understand that which genes are making it resistant to disease, which genes are making it resistant to drought also. So only by doing genome sequencing it is possible. This technique helps in, so this statement is correct, so the first statement is correct. So if you look at, if the first statement is correct, so this is wrong. So you're left with you have to check for 3 and you have to check for 2 also. So this technique helps in reducing the time required to develop new varieties of crop plants. So if you have a good knowledge of the entire sequence of the plants, then we can very quickly develop new varieties of crop plants also. So this is also true in this case. It can be used to decipher the host pathogen relationship in crops. What is this host pathogen? So some disease are able to attack the crop plant. Why they are able to attack crop plant? Because presence of some genes. So this linkage between the pathogen and the host plant, can be established by sequencing only. So this is also a correct statement. So what is the answer here? So the answer is D in this case. Let us look at this GAC. So GAC, you must remember that it is a statutory body. It is a statutory body, and it is under Ministry of Environment, Forest and Climate Change. And there are rules for manufacture, use, import and export of storage of hazardous microorganisms, genetically engineering organisms or cells, 1989. So these are also known as your 1989 rules. So it follows these 1989 rules, and it is a statutory body formed under the EPA, that is the Environmental Protection Act. That is the Environmental Protection Act. What are the different roles? The different roles are what, to give permission for field trials, to give final approval for GM crops. If there is a breach of the laws or rules, then it can take punitive actions also. So these are some of the functions of the GEAC. GEAC. So what we'll do, that we'll also look now at space technology. So in space technology, so let us try to answer this question, and then we'll move to other parts of space technology. Yes, give it a try. Yes, anybody? Are you all giving prelims this year? Aditi says A, and along with A, not sure though, C, 1 and 2. C, 1 and 2. Okay, so let us just read this question. PSLV launch satellites useful for Earth resource monitoring, whereas GSLVs are designed mainly to launch communication satellites. So, so yes, this statement is correct. So this statement is correct, that it is used for Earth resources monitoring. And GSLV, so GSLV are designed mainly to launch communication satellites. So that was the purpose of the GSLV. So this statement is correct. What about the second statement? Satellite launched by PSLV appear to remain permanently fixed in the same position in the sky as viewed from a particular location on Earth. So this statement is not correct. Why? Because PSLV PSLV means they will be sending satellites to polar orbits. So the satellites will be appear to be moving in north south direction, and polar orbits are your low earth orbit. Polar orbits are your low earth orbit, and because they are low earth orbit, so they move very quickly around earth, so they would be making multiple rounds around earth. If you talk about bodies, so they can even make up to more than 16 times rounds, or 16 rounds around the earth in a day. So it cannot appear to be permanently fixed. So PSLV or polar satellites, it cannot appear to be permanently fixed in the sky. If you want to send it, or if you want to make it fix in the sky, then you what we do is that we send it to what are known as high earth orbit or geostationary orbit. So geostationary orbits are 36,000 kilometers above the surface of earth. So if we are sending any satellite to this type of orbit, then it can remain stationary, but in polar satellites, they are not stationary. So your second statement is wrong. So now let us look at this statement. GSLV Mark III is a four-stage launch vehicle. GSLV is three-stage. GSLV is three-stage. So first stage is solid, then second stage is liquid, then the third stage is cryo. Whereas PSLV has four stages. So it has four stages. So it becomes with solid, then liquid, then again solid, then again liquid. So what UPSC has done is that they have transposed it. Had PSLV been written here, then this statement would have been correct. But because it is written GSLV here, so GSLV is three stages only. GSLV is not four stages. So this statement is also correct. So the correct answer in this case is your one only. But this question would have suggested to you that we need to know these launch vehicles in some detail. We need to know the launch vehicles in some detail. So we'll try to understand these launch vehicles. But in order to understand these launch vehicles, we also need to understand the basic working mechanism of these launch vehicles. So how do we design these launch vehicles? So how we move them? So what is needed is we need some mechanism. So in this we need to understand what is a rocket engine, or how do rockets move. We first need to develop a basic understanding of rocket engines, and then we'll try to understand this SLV. Then we came up with ASLV, then we came up with PSLV, then we came up with GSLV, and now a parallel development is going on, that is we have developed SSLV, and we are also trying to develop what is known as a reusable launch vehicle technology demonstrator. So we are trying to develop so many type of launch vehicles. So this was the main sequence of development, but we are seeing some other parallel developments also. So from PSLV we have moved to GSLV, but for very small satellites we are trying to develop this SSLV, and this is going in trial phase. So is this will be the reusable one. We'll see the importance of the reusability criteria, but first we need to understand the basic design of a rocket engine. So what you're seeing here is that this is the payload. This is the payload is in this case is your, let us say, a satellite. Payload is a satellite, so you have kept a satellite here, and this is what is known as payload fairing. This is known as payload fairing. So payload fairing is the covering of the launch vehicle. So in this payload fairing only the satellite is kept. So let us say the satellite is here, and then you have the guidance system. So now the main part is that it is containing fuel and is containing oxidizer. See, this rocket will go in space. Because it is going in space, it will have no supply of oxygen. No supply of oxygen. So no, since there will be no supply of oxygen, so how will the fuel burn in space then? So in order to burn the fuel in space, we carry the oxidizer. So we have the oxidizer also, we have the fuel also, and you can see through pipes. Through pipes we are bringing the fuel and the oxidizer, and this we are burning. Through pumps we are bringing, and we are burning it in the combustion chamber. So there is this combustion chamber. In the combustion chamber, the idea here is that we need to produce what? Hot exhaust gases. So if we are able to produce these hot exhaust gases, then we'll be see how do rockets move. So rockets, the design of the rocket is very similar to, let us say, this is a balloon. This is a balloon that you have filled with air. So if you have filled the balloon with the air, and now if you leave this end open, so what will happen when you leave this end open? So the air will move out in this direction, the balloon will move in this direction. So what we are doing in a rocket is that we need to produce hot exhaust gases here. So we are producing hot exhaust gases here. Those hot exhaust gases, they are made to move out in this direction. Because they are moving in this direction, it is producing thrust in equal quantity in opposite direction. So this principle of reaction and action and reaction is your Newton's third law of motion. So rockets work on Newton's third law of motion, that every action has equal and opposite reaction. So this rocket, you are seeing that there is a fuel chamber, there is an oxidizer chamber, and the fuel and the oxidizer, with the help of pipes and with the help of pumps, it is brought to the combustion chamber. They combine in the combustion chamber, and a lot of hot exhaust gases are produced. So that is how the thrust is produced. So are you able to understand this part, that how we are able to produce thrust in rockets? Because we are able to produce hot exhaust gases, these hot exhaust gases are made to pass out through the exhaust nozzle. So because of that, we get a thrust in the opposite direction. So this is how rockets are designed. So you would always keep hearing these terms like what is cryogenic. Cryogenic. What do you mean by cryogenic engines? Yes, what do you mean by cryogenic engine? Yes, anybody? Cryogenic engine is in news. Why? Because in India there is this private space startup, which has made the world India's first private cryogenic engine. Private cryogenic engine. So what is cryogenic? And they have also gone for 3D printing. Hydrogen fuel use. So hydrogen, what do you mean by cryo? What do you mean by cryo, first of all? Yes, we are using hydrogen. In what state? In what state are we using hydrogen? Yes, so at very low temperature. So cryogenic is derived from Greek word cryos. Cryos means frost. So cryos means frost. So in science, when we say any engineering that operates below, so below minus 150 degrees Celsius, any process or product that are made below minus 150 degrees Celsius, we call it as cryo state. So what are we keeping at cryo state? We are keeping the fuel at cryo state. So fuel is at cryo state. No, we'll see. It is not at gaseous state. At this oxidizer is at cryo state. So if you keep it, or if you take it to very low temperatures, that is to minus 253 degrees Celsius, this gaseous hydrogen actually converts into liquid hydrogen. And uh, and your gaseous oxygen below minus 180 degrees Celsius, it converts into liquid oxygen. So what are we doing here? So because of going of at cryo state, the volume of these gases, they reduce drastically. So it is said that the volume of the hydrogen will reduce around 400 to 500 times. 400 to 500 times. Similarly, liquid hydrogen also will reduce to some multiples of hundreds only. So what is happening here? Because the volume is reducing, the energy density is increasing. So because of reduce in volume, the energy density means the per unit volume area, we will see that there is more hydrogen. So per unit in this area, now if this was the normal gaseous state, so you will see one or two molecule, but in this you will see what, millions of molecules. So because of that, what is happening, the energy density has increased. So because the energy density has increased, so this liquid hydrogen when this liquid hydrogen and liquid oxygen, they burn in this combustion chamber, so they will be producing a lot of hot exhaust gases. Because they are able to produce a lot of hot exhaust gases, so they will produce a lot of thrust. So they will produce lot of thrust because they are able to produce lot of thrust. So that is why it is said that cryogenic engines are very very powerful engines. Why? Because the because the energy density has increased drastically. Now can you tell what is semi cryogenic? What do you mean by semi cryogenic? Or I'll write on this side. What do you mean by semi cryogenic? So semi-cryogenic was in news. So HAL has delivered the heaviest semi-cryogenic propellant tank to the ISRO, replacing L2 in existing Mk2 launch vehicle. So cryogenic. So what we are using here is that refined kerosene is used instead of liquid hydrogen. Refined kerosene is what liquid at room temperature itself. It is liquid at room temperature itself. So we are saving on the cost of what going for cryogenic cooling of hydrogen from gaseous hydrogen to liquid hydrogen, that is not required. So we are able to save on this cost. So we are able to save on this cost. So this is not required. So why? Because the kerosene is a liquid at room temperature itself. So that is the advantage that we have. Now refined kerosene, which is lighter than liquid, can be stored in a normal temperature. So it is more powerful, environmental friendly, and more cost effective. So what you are saying that the fuel is at room temperature only, liquid, and you are using this as this is at cryo. So this is at cryo. So that is the difference in semi cryogenic. So your oxidizer is at cryo but not the fuel. So this is known as semi cryogenic engine. Now let us come back to the start of the launch vehicles in India. So in India we started with this launch vehicle. So this launch vehicle is your SLV. This launch vehicle is your SLV. But the problem with SLV was that the payload capacity, this you can see when you say payload capacity, this you must understand that this is the carrying capacity of the launch vehicle. So the payload capacity was very less. So with this program we started in 1970s, and we felt that this has very low capacity, so we switched to ASLV. ASLV is augmented. It is known as augmented. This is known as augmented launch vehicle. Augmented SLV is satellite launch vehicle. ASLV is augmented satellite launch vehicle, and these two are not in service. These two are not in service. So we utilize what? We utilize this PSLV, GSLV, and GSLV also you are seeing, we are seeing that evolution. So we are using PSLV, we are using GSLV. So in PSLV you can take a note that in the first two generations we are using only solid fuels. We are only using solid fuels. For the first time we used liquid fuel in your PSLV. So we have used the liquid for first time in PSLV. And PSLV is what? 4-stage rocket. PSLV is 4-stage rocket. So PSLV you can see PS1 we call it as the first stage. So this is your first stage, this is your second stage, this is the third stage, and this is the fourth stage. So the first stage is, it needs what? Very high thrust. Because it needs very high thrust, and why it needs high thrust? Because of the liquid, because of the lift of mass is very high, so it needs high thrust. So to aid the first first stage, we have what is known as the strap-on motors. So we have 6 solid strap-on motors that augment the thrust provided by the first stage in its PSLV. Then you have the second stage. The second stage is the liquid stage. So for the first time we made liquid engines and we deployed it in our PSLV. The third stage is again the solid stage, and the fourth and the final stage is we are using liquid engines only. Liquid engines for propulsion. And then what you're seeing, this part is called as your payload fairing. This part is called as payload fairing, that is the covering of the rocket. This part you would see that this part will eventually fall off in the ocean. They fall off in the ocean. Inside this, you, let us say you have the satellite. So what happens that these are your different stages. So this is first stage, this is second stage, this is your third stage, this is the fourth stage. So what happens that after some time, so after few seconds, this stage, first of all, these strap-on motors will fall off. Strap-on motors will fall off. Then this will separate, and this will fall off in the ocean. Then this part will switch on. This part will run the vehicle for some time. Then eventually this part will also fall off in the ocean. Similarly for this part, and similarly for this part also. So because of that, we say that our PSLV and GSLV are expendable. It means that they get wasted away. Because of the design. So what we want that somehow we should be able to reuse these important parts and refly them again. So we will see that how Elon Musk is trying to do that. So he believes that if humans have to be sent to Mars, he believes that Mars will be the insurance for humans, and if you have to send humans to Mars, we would need cheaper space travel. So for cheaper space travel, it means that our launch vehicles should be robust and they should be cheap also. How we can make these cheap by making it reusable. So India is also trying its hand at the reusable launch vehicles, that we'll see a little later. But with respect to PSLV, it has emerged to be what is known as the workhorse of ISRO. It has emerged as workhorse of ISRO, and it has sent more than 200 foreign satellites, and it has sent more than 50 Indian satellites. And from 1994 it had a very good streak. From 1994 to 2017, it sent consecutive 34 successful flights. So the success rate of PSLV is very high. We have used PSLV for Chandrayaan 1 mission, for Mangalyaan 1 mission. So it has been used in some of the marquee missions also. That is why ISRO considers it as the workhorse. So have you seen that movie Rocketry movie of R. Madhavan? Yes, anybody? Hmm. So if you get time, you should watch this rocketry movie. So why this movie is good? That you will be able to understand that what was the contribution of Professor Nambi Narayan. So Professor Nambi Narayan and APJ Abdul Kalam, they were students of Vikram Sarabhai, or they were proteges of Vikram Sarabhai. So Vikram Sarabhai is the person who started India's space program. So Vikram Sarabhai, so he developed these two persons mainly. So what he did is that he encouraged Professor Nambi Narayan to become expert in liquid engines. So expert of solid engine was Dr. APJ Abdul Kalam. But Professor Nambi Narayan realized that if India wants to increase the payload capacity of its launch vehicles, then it will need to develop a more sophisticated engine that uses liquid engines. So he went to Europe and he tried to study from the different professors and universities, and he came up with a design of liquid engine. So it was because of his contribution that India was able to develop the liquid engine. That liquid engine is called as, so here you can see the name, it is called as Vikas. Vikas is because of, it is a combination of two words, Vikram Sarabhai. So he named in honor of his guru, that is Vikram Sarabhai. So he named this engine as your Vikas engine. So this engine is still used, and is still used in your PSLV. It is also used in your GSLV, but for the first time this liquid engine was used in PSLV, and it was the contribution of Nambi Narayan only, that he wanted to develop the cryogenic engine in India. But because of the breakdown of USSR, India's attempt to develop the cryogenic engine was delayed. So in India, he was the pioneer in cryogenic engine only. But somehow, if you watch that movie, you'll come to know that because of some global conspiracy, he was put behind bars. So because he was put behind bars, he was not able to contribute to the development of the cryogenic engine. So that is why the development of the cryogenic engine in India has taken so long. It took around 20 years for India to develop indigenously the first cryogenic engine. So now what you are seeing, that even the some of the private space startups, they are able to develop the cryogenic engine. So tomorrow we'll see that what is this GSLV and how many stages are there in GSLV. But PSLV has proved to be really a workhorse for ISRO. So but PSLV had limitations, or it has limitations, that the payload capacity drastically improved from, in this slide you can see. So the payload capacity, you can see that payload capacity of the ASLV was 150 kg. Here you can see the payload capacity more than 10 to 11 times, more than 10 to 12 times. The capacity increased because of use of liquid engines. But even this capacity is not sufficient to send something like the some of the heaviest telecommunication satellites, such as the GSATs. Now to send GSATs, we needed some very powerful launch vehicles, and then we came up with what is known as GSLV, and we are in GSLV for the first time. We are using cryogenic engine. In which stage are we using cryogenic engine? So we'll see. Okay, so we'll see you tomorrow. If you have any doubt, you can go ahead. Yes, are there any doubts?